I want to thank everyone for coming today. Today is our virtual Lunch and Learn. It's a microbe or micro view of prairie restoration. My name is Ellen Holste. I'm the community program manager at Cedar Creek Institute. Today we have Kathryn Docherty, I apologize, from Western Michigan University, and she's going to talk a little bit about prairie restoration today. I'm going to turn it over to Kathryn, but I have a question for Kathryn first. Since we're talking about microbes and prairie restoration, I would be interested in knowing, how did you get interested in microbes, Kathryn? Oh, and I need to unmute you. I apologize. 1 second. Oh, you are unmuted.
Okay. Can you hear me?
Yep, we can hear you.
All right. How did I get interested in microbes? That's a great question. I started becoming interested in microbes, I think, during college. I went to a small liberal arts college in upstate New York. Actually, you can see a painting of the Hudson River behind me, which is by my mother, who's actually in the audience. Thanks, Mom. And so I grew up along the Hudson River and then went to college near the Hudson River. And it was during this time when the ecosystem was really being affected by PCBs that had made it into the river. And there were all these environmental impact statement discussions going on. In the meantime, I'm learning about how microorganisms can be used to clean up pollutants and really are benefits to ecosystems. So kind of that context of this beautiful ecosystem and working on ways to naturally protect it really inspired me to start pursuing microbial ecology. My PhD work was at the University of Notre Dame, looking at how to biodegrade organic pollutants with microbes. And then since then, I've branched out into the sort of the opposite side of the question: how are the things that humans do impacting microbial communities? And what I'm going to talk about today does have a bit to do with both of those sides of microbial ecology. So I can go ahead and introduce my talk. So I am a faculty member at Western Michigan University in the Department of Biological Sciences. And I do want to thank everyone for attending and for the Cedar Creek Institute for hosting the webinar, and Ellen for organizing everything. Thank you so much. I was scrolling through the list of participants, and it looks like we've got a really broad variety of audience members. Some of my colleagues from Western, as well as at Kellogg Biological Station, are here. There's some undergraduate students and faculty who are participating in the Cedar Creek Institute summer research activities, as well as some looks like conservation volunteers and other people who are in the community and are interested. So thank you all for coming. I hope that my talk gives you something interesting to think about during this quarantine time, but also I'm going to pause throughout and give you the opportunity to type a question into the chat if you have it. So if there's something you want me to explain further, I'll pause and you can add a chat question. So as I said, I'm an associate professor at Western, and I'm a microbial ecologist. We have a lot of different projects going on in my lab right now, but today I wanted to focus on one that is one of my favorites, and it has to do with one of my favorite systems that we're working in currently. And it's a system that has the potential to provide a lot of really great ecosystem benefits to Michigan and to other agricultural areas. And so I also want to mention that I'm sort of an amateur photographer, so I really enjoy taking photos, especially of some of these research sites. So all the photos in this presentation are my own, unless there's a photo credit listed for someone else. Technical difficulties, 1 minute. All right, there we go. So this is this ecosystem, this prairie conservation corridor that you can see planted within a cornfield. And this particular corridor is located at the Edward Lowe Foundation, which is near Decatur, Michigan. And if if you're from Michigan, you're familiar with the mitten. Decatur is about here in Southwest Michigan. This particular corridor was planted in 2014, and it really is obvious when you look at this agricultural field that there's this highly diverse strip of prairie plants within this agricultural ecosystem, and it really stands out from the surrounding cornfields. A lot of times farmers are encouraged to plant these types of native plants in marginal lands and try to increase plant diversity in areas that they're not necessarily using for cropland. But as of the 2018 United States Farm Bill, conservation corridors like this one are actually also included into the Conservation Reserve Program, where farmers can apply for some funds back if they plant these types of corridors in their actual agricultural field. So as you can see, this one is actually cutting across usable farmland. And the reason for this is depending on where these corridors are placed, they can really have a lot of benefits to the surrounding ecosystem. And I'm going to talk about some of those benefits as the first part of my talk. But depending on where they're placed, they can really improve particularly soil health. However, placing these corridors into an active agricultural field obviously comes at a cost. This farmer is no longer able to plant this area with their crop, so they have to have some sort of major benefit, an ecosystem benefit that is going to outweigh the profits that they might have made with the crop that they would have planted. Most of the time, farmers and land managers manage these types of conservation corridors by planting a native seed mix that has a diversity of different native plants, and then manage it using above ground strategies that focus on burning or invasive species removal, but they're usually focused on the very visible aspects of managing this type of ecosystem. So part of what my lab is doing and what researchers in other labs are doing throughout the country is examining ways for optimally managing agricultural ecosystems to make sure that farmers and managers who place these types of management strategies into active cropland are getting the best optimal benefits for the ecosystem, both above ground and below ground. So I want to maximize those benefits of these conservation efforts. So in terms of an outline, I'm going to talk about what are these specific benefits of prairie conservation corridors that we know about so far. Then I'm going to get into really what the title of my talk was about, is why soil microbial communities matter at all in this system. And then finally, I'm going to talk a bit about integrating soil microbial community management strategies into the regular management strategies that are already in place. So what are some of the benefits of prairie conservation corridors in agricultural systems? Well, first of all, and most obviously, they increase plant diversity. This is one of my undergraduate students walking through one of these corridors at the Edward Lowe Foundation. You can see the grass is taller than she is. She's about 5'8". And all the plants are taller than she is. There's a huge amount of diversity in this prairie plantation. And you can see that it's a sort of massive amount of above ground biomass that has accumulated in these systems, and it's a perennial system. So that biomass comes back every year and then senesces and then decomposes and then rejuvenates in the spring each year. So that plant diversity is accompanied by all kinds of other benefits. First of all those plants serve as potential sources of pollen for pollinators. So it provides a source of pollen as well as a source of genetic diversity for these plants. It provides a habitat for pollinators that might not always be present. And by providing a diverse habitat for pollinators, you can also provide for diverse types of pollinators. So many pollinators are becoming threatened or endangered because the specific plants they require aren't present. By having a diverse plant community, you can diversify pollinators. It also improves habitat for animal species that require conserved grassland areas as their habitat. So birds and other animals that require these types of habitats have a habitat within an agricultural ecosystem that they might not have had without it. Another major benefit of prairie conservation corridors is, of course, thinking about migratory species. So one of my favorite migratory species is the monarch butterfly, which is why milkweed is one of my favorite prairie plants, in answer to Owen's initial question. It's because monarch butterflies require milkweed in order to undergo their 3-generation life cycle each year and be able to feed and reproduce. So having this specialist type of species that requires this one type of plant is really dangerous for that organism, because if that plant is no longer available in large quantities, then that migratory organism may not be able to make it through their migration period. So by planting these diverse types of prairie conservation corridors inside an agricultural system, you provide that habitat while still maintaining the ability to produce crops and farm. Finally, some research has shown that with all these benefits of increased plant diversity, increasing the types of pollinators and the types of animals, and the types of migratory species that might go through a system, that results actually in a protection for the surrounding crops that are adjacent to these prairie conservation corridors. In some cases, you will see fewer crop pests. So for example, crop pests like aphids might be outcompeted by some of these other insects, or they might be predated upon by some organisms that, like birds, that are living in this habitat. So by providing habitat for other types of organisms in the ecosystem, it naturally protects the crops against certain types of pests, and potentially reduces the need for pesticides. All right, so those are the above ground reasons why prairie conservation corridors are cool and neat ecosystems, and a nice thing to include in an agricultural management plan. But all of that is based on what's going on in the soil. And one of the most obvious things that obviously occurs when you plant prairie plants into a agricultural field is that those prairie plants have much deeper root structure. So this is a photo of a corn seedling and a big bluegrass seedling, or a big blue stem seedling, sorry. And you see that the root structure, even in these two seedlings, is just very different. So corn is on the left and big blue stem is on the right. And you see this root structure for big blue stem is much more extensive than the root structure for corn. In fact, if you look at this diagram that is from the USDA Illinois Native Plant Guide, you see that Kentucky bluegrass, all the way on the left, is the typical grass that you might have in your lawn, and you can see that its root structure is really fine. So you can easily remove Kentucky bluegrass from your lawn because the roots don't go very deep. However, if you look at some native types of plants that you might see in prairie conservation corridors, you see that root structure really extends very deeply into the soil. So you get down to 15 feet deep with some of these prairie plants. And that has an overall effect of both increasing the amount of plant biomass that is below ground, which becomes important for the amount of carbon that is maintained in the system. But it also becomes important for the problem of erosion. So farmers today are facing huge problems with soil erosion, especially as we continue to see greater flooding events and then followed by drought events. So these really extreme flood events are becoming more and more prevalent. And when an agricultural field floods like this one, it leads to loss of soil and soil erosion. So a lot of that sediment gets washed away. There's a group out of Iowa that is studying prairie conservation corridors, and they've shown some really neat results that suggest that depending on where a prairie conservation corridor is placed in an agricultural field, it will reduce water runoff by almost 40%. It leads to almost 20 times more soil retention, and also leads to about 4 times more nitrogen and phosphorus retention. So if these prairie conservation corridors are placed just in the right area where runoff may occur, then these major benefits to farmers can also be implemented. So the amount of money that a farmer is spending on maintaining soil health or fertilizing a field might be outweighed by placing a corridor and not planting a crop in one particular area because it might protect the field from runoff. So this brings me to what this entire ecosystem is based upon. The entire ecosystem is based upon the soil that it's planted in, right? So everything I've said so far results from these traditional management practices where a land manager or a farmer will buy a seed mix and then they'll plant those seeds into their agricultural field, and then they'll use above ground approaches to manage that system. And then they're looking at this success of the prairie conservation corridor based on visible elements. So what they can see. Do they see greater plant diversity? Do they see less soil erosion? So what we think about in my lab group is what is happening with the invisible elements of the ecosystem, the microbial elements of the ecosystem. The soil is a structure upon which all these agricultural ecosystems are built. And soil microbial communities are really the engineers of that ecosystem. Without those organisms, this ecosystem isn't complete. And they're every bit as important as plants and animals, but because they're invisible, they're just far less understood. So there are, in my mind at least, two different groups of organisms that are microorganisms that inhabit the soil. The first are those that form really direct associations with plants. And some of you mentioned that arbuscular mycorrhizal fungi are your favorite types of microorganisms. And I agree, they are definitely microorganisms that form associations with plant roots. These are fungi in the soil that extend the root surface area in the soil. So this is a photo here on the left of a plant that has been associated or is associated with mycorrhizae, and it's this symbiotic relationship between the fungi and the plant that results in a greater extension of root area by these fungi that produce these tendrils of hyphae that extend into the soil, and then they can interact with nutrients and water supplies that the plant roots might not be able to access. In the meantime, the plant is photosynthesizing and it's producing chemicals through the process of photosynthesis that it then releases into its roots and can be used by the fungi. So this symbiosis leads to benefits for both the fungus and the plant. And these associations are really common with prairie plants. Another direct association is between plants and the bacterial group Rhizobia. Rhizobia are nitrogen-fixing bacteria that they will take nitrogen from the atmosphere and fix it into a form that can be used by plants. And so these plants have adapted to create these little structures, these nodules in their roots that you can see in this picture on the right, that provide a habitat for these nitrogen-fixing bacteria and Rhizobia. And in the meantime, the Rhizobia are then able to fix nitrogen and provide that nitrogen to the plant. So again, another direct symbiosis between microbes and plants. And prairie conservation corridor plants also may associate with Rhizobia. The other side of soil microbial ecology is not just the direct associations with plants, but the soil microbial communities themselves. So the microorganisms that are present in the soil are co-inhabiting the same ecosystem as all of these plants. They may not be directly associating or in a symbiotic relationship, but there are millions of microorganisms in each gram of soil. Not just each cubic foot of soil, each gram of soil has millions of microorganisms. And all of them provide some sort of ecosystem context for the soil, for the plants, and are responsible for very important processes. So they may be also using these plant root exudates that plants produce. They are involved in decomposition of plant material, where they're breaking down that plant biomass each year and converting it into more useful forms of carbon. In that sense, they are involved in the global carbon cycle and in storage of carbon in the soil versus production of gases that are related to decomposition. Microorganisms are also involved in nitrogen cycling. We already talked about nitrogen fixing bacteria, but there are also organisms that are nitrifying and denitrifying in the soil and mineralizing organic matter and producing nitrogen that's useful to plants. And all of this leads to this potential for soil microorganisms to either produce or consume a bunch of different greenhouse gases. So carbon dioxide, methane, and nitrous oxide are all important greenhouse gases that are produced through microbial processes, but also can be consumed through microbial processes depending on which particular microorganisms are present. Right, so what's the problem? If you've bought all this so far, so prairie conservation corridors are important and microbes are also important. The problem is that tilling, fertilization, and monocultures, which are these traditional agricultural activities, have very long-term effects on soil microbial communities. So all of these factors that are involved in soil microbial ecology are affected by tilling, fertilization, and then monocropping for many years. And the problem is that it takes about 3 decades to recover soil bacterial communities, if at all, and this may also be the case for fungi. So this problem is potentially disastrous when you start to think about trying to include and maximize the benefits of a prairie conservation corridor. So with traditional restoration practices, it actually may not be possible to completely recover all soil microbial functionality and carbon storage, and it may not be able to recover the entire ecosystem benefits that soil microbial communities can provide without some more below ground focused restoration approaches. So one way to determine whether a community has recovered from agricultural activity, and how do we know that it takes potentially 3 decades, if at all, to recover? One way to do that is to examine what's called a remnant. So you can imagine a little parcel of cropland that just was never planted with any agricultural crops. And these are called remnants. They're few and far between, especially in Michigan. They're hard to find, and they're usually in kind of odd places. But this is a traditional practice that folks who study plant and microbial ecology in restored systems use remnants as bases of comparison to determine whether a community as it's being restored has recovered to the point of what a remnant community looks like. So in my group, we use genetic approaches. If you were a plant ecologist, you could go outside and look at the plants and identify them potentially visually and count how many of them there are. In my group, we can't do that because there are millions of microorganisms in the soil. And so we rely on genetic approaches to do this type of community-based sampling. So we go out and we collect soil samples, and then we extract the DNA from that soil sample and look at a very specific marker gene that tells us what type of bacteria is present. It's called the 16S ribosomal RNA gene. And it's a marker gene that allows us to determine what taxonomies of organisms, microorganisms, are present in soil. And since this data set you could imagine is going to be really complicated, if you have the abundances of many different types of organisms, up to millions of different sequences, it becomes really complicated to look at if you wanted to make something like a pie graph, for example, right? So if you're thinking about the demographics of a city, you could easily look at that in a pie graph. When you start to get to millions of different types of species of bacteria, you need to do something a little bit different. So what we do is we use these statistical approaches called ordinations that create condensed data points for each community. So we can examine the relatedness between 2 communities based on a two-axis system, and that's what this graph is showing. So the basics of this ordination are that these two axes are separating out our communities based on what organisms are in them. And if you look at the two gray triangles, those are two different soil samples from two different remnant prairies, and they are fairly closely related to each other. So the 2 points are close to each other. That means those two bacterial communities are really pretty similar. Compare that to the blue square, which is active cropland. And that blue square is actually really far away from these gray triangles. So what that means is that that community that is represented by that blue square is not similar at all to the community that is represented by the gray triangles. All right, so we know that cropland bacterial communities and remnant prairie bacterial communities are really different. Well, what if we restore and we look at restorations over time? We looked at a restoration that occurred in 2017. These data were taken in 2019, so it was a 2-year restored prairie. And you see that these two red dots are the communities that represent, are the points that represent the communities from these two different restorations that occurred in 2017. And those are more similar to the cropland bacterial community than they are to the remnant community. So not much has changed, a little bit of a shift towards what a remnant might look like, but it's pretty similar to a cropland. What if we go further back in time in restoration? So looking at a restoration from 2008, from 2002, and then even back 30 years to 1990, what we see is there's this slow progression across this continuum, this chronosequence of prairies, where by 1990, that community is starting to resemble the communities that are present in the remnant prairies. So it's taken 3 decades to get there, and it's still not quite there either. So that 1990 community is not exactly on top of where those two gray triangles are located. But what we see is this slow progression over time towards some similarity with these remnant prairie bacterial communities. This is where we get this idea that it takes at least 3 decades to recover, is these types of chronosequence studies that look at restorations from different ages. And we've shown this in Michigan, and this is a small data set compared to a couple of other studies that have shown it also in Illinois and in Kansas, is that there is this really slow progression. It takes at least 3 decades, if at all. And all of these different points in between that 30-year period are these transitional communities that are slowly changing towards these communities of bacteria that might look like remnant bacteria. So in the next part of my talk, I'm going to get into, well, why do soil microbial communities matter in conservation corridors? It seems like that these plant communities establish, but you know, what benefits can restored soil microbial communities provide? So one of those benefits has to do with soil carbon storage, and another benefit has to do with habitat for late successional plants. But I'm going to pause here and see if there are any questions in the chat, and then I'll go into some of these details.
Kathryn, there's a few questions.
Okay.
One question that Kenneth had was, does historic corn plants found in North America prior to cultivation in monocultural fields by European descendants have a more extensive root structure?
Do they? Is that the question?
Did they? So did historic corn plants prior to cultivation in monoculture fields, did they have more extensive root structure than I believe today, is what he was asking.
Oh, that's a good question. I don't know the answer to that.
Okay, we'll get back to that one.
If you want to email it to me, I can take a look at it. But
another question we had is there research showing that the diversity, I think they're referring to plant diversity, reduces crop pests more than chemicals typically used?
Comparatively, that's a good question. I don't know the answer to that one either. I do know that crop pests decrease when there are prairies surrounding an agricultural field. I don't know directly if you did this comparison of applying a bunch of pesticide to one field that has no prairie around it versus a system that has some prairie around it. If anybody has done that type of work, I would guess it would be Doug Landis's group at MSU. So that might be where we might find the answer to that question.
All right, another question we had was by Jeff. What microbial constituents are missing in a 30-year-old prairie restoration?
We're going to get to that in just a minute.
Good question, Jeff. All right, and then one more we had. Could it be dangerous to draw sensitive pollinators to conservation corridors that exist among conventional farm fields that use pesticides? Or is it better to give them the habitat and hope they aren't killed off by pesticide use?
That's a really good question. I would say it's better to give them the habitat and hope they're not killed off, and hope that you can decrease the pesticide use as a result of having this native habitat present. But that is a really good question. If you're drawing these potentially threatened organisms to this area where they could get killed, are you actually not benefiting them? I think then it's important to start to establish prairies in other places where pesticides aren't used. And one of the things that I will mention is that it doesn't just have to be agricultural systems where these prairie plants are planted. It can be in suburban systems or at business parks. Western has a really excellent example of integrating prairie type native plants into their engineering campus. So I think not just agricultural systems have to be involved in promoting pollinator abundance and habitat.
Let's keep going.
Sure. All right. These are all great questions. Thank you. If I don't know the answer, I'm going to share my email at the end, and we can always email back and forth, and I can check out and find an answer for you. All right. So why do soil microbial communities matter in conservation corridors? And the first reason has to do with soil carbon storage, and that gets at the question of what's missing. So this is a map showing where the tallgrass prairie ecosystem once extended prior to agriculturalization of the Midwest and Central Plains. So this extensive ecosystem ran from all the way from Canada down to Texas, and most of Illinois and Iowa were covered in tallgrass prairie, and it actually led to development of a very particular soil type in that area that is rich in organic matter and is excellent for farming. Today, that tallgrass prairie ecosystem is located here, mostly in Kansas, a little bit in Nebraska, and most of the extensive ecosystem is now converted to cropland, which again, that's the reason why, is because the soil type was excellent for planting crops because of this long-term tallgrass vegetation that provided a really rich organic soil. When this occurred, it's estimated that there was a loss of about 993 teragrams of soil carbon from the soil. So this really rich organic soil was now suddenly tilled and cultivated, and the biomass from the tallgrass prairie was no longer senescing and making its way back into the soil. And as a result, that led to a huge loss of carbon, most of which was converted to carbon dioxide, which is a greenhouse gas. So when we think of agricultural ecosystems contributing to climate change, this is one of the reasons, is because this conversion of soil carbon to atmospheric CO2 is a major flux from what once was this tallgrass prairie ecosystem. There are other greenhouse gases that are involved in agriculture as well, methane and nitrous oxide being the predominant ones, but all three of them are the result from agriculture as well as many other anthropogenic activities. So why did this occur? We think it's because there was a change in this long-standing perennial biomass, first of all, but that led to a change in soil microbial community composition, and particularly a shift from what are called oligotrophs to copiotrophs. So what are copiotrophs? Copiotrophic microbes are those that are often predominant in today's agricultural systems. And copiotrophs don't have to just be microbes. It's a character trait of any type of organism. But this is an organism that's adapted for disturbance. It reproduces quickly and in high abundance, but it also dies fairly quickly. And that biomass can then be broken down and is often converted into carbon dioxide. So agricultural systems are characterized by having more bacteria, and particularly more bacteria in the phyla, proteobacteria and actinobacteria, which are thought to be these copiotrophic groups of organisms that have these different ecological traits, as well as reduced numbers of fungi. So those arbuscular mycorrhizal fungi are not adapted for disturbance. If you can imagine tilling through an agricultural field and removing arbuscular mycorrhizal fungi might disrupt their habitat and reduces their abundance. So to give you an idea of a copiotroph that you're all familiar with that's visible, you might think of this guy. So this is your characteristic copiotroph. It's an organism that is adapted for disturbance, and no matter how many times you mow your lawn, this organism is going to come back, and it reproduces very quickly. It also dies really quickly, and is fairly difficult to get rid of because it's adapted to this disturbance ecosystem, dandelion. So the dandelion microbes are these proteobacteria and actinobacteria. So what used to be in these soils before conversion to agriculture? Again, this tallgrass prairie ecosystem once extended this entire length, and it's a really neat study that was conducted and published in 2013 that showed an estimated model of what types of microorganisms used to be present in this system. And what they think used to be present in most of this tallgrass prairie ecosystem are this phylum Verrucomicrobia. And Verrucomicrobia is not well understood. It's a phylum that we didn't really even recognize as being very prevalent in soils until the last 5 to 10 years or so, based on sequence information. And we don't really have that many cultured representatives of Verrucomicrobia. But what we do know is, based on genetic techniques and genetic inferences, is that these types of microbes are oligotrophs. They don't do well with disturbance. They reproduce pretty slowly. They produce a lot of cellular biomass in the form of their cell walls or their cell membranes, and a lot of lipid biomass, leading to microbial carbon storage. So carbon is stored inside the biomass of that organism. They also are larger. So fungi, for example, are just larger cellularly and produce larger structures than bacteria in the soil might. And they also, if you're thinking in terms of plants, they often are those that live an entire season. So oligotrophic microbes used to exist in prairies. So we think there used to be more fungi. There used to be more of these Verrucomicrobia and more of the phylum Acidobacteria, all of which have been classified as oligotrophs based on these types of genetic approaches that predict these types of character traits as opposed to copiotrophic traits. So if we're thinking about a tractable example of an oligotroph, we might think about this rattlesnake master. So this is rattlesnake master, very difficult to cultivate. Doesn't like to grow too well in a disturbance area. It grows to over 6 feet tall. This one's in my front yard, and I had to hold my arm up above it to get a picture of it. It produces a lot of biomass, much more than a single dandelion plant would, but it also reproduces really slowly and doesn't produce nearly as many seeds as a dandelion. So this is maybe a characteristic of an oligotroph. So if you think of oligotrophic microbes, you think of those that have these types of character traits. All right, so at this point we know that conversion of prairies reduced the amount of oligotrophs in the soil, so it was lost habitat, meaning there's fewer fungi. It also was a changed habitat, which led to fewer oligotrophic bacteria. We also think from a few different studies that it takes at least 30 years, if ever, to restore soil microbial communities using these traditional above ground restoration practices that focus on plant diversity. And this kind of results in something that's an example from the oldest restoration in the country. It's in Wisconsin, that even after 65 years of restoration, there's more respiration of CO2 from restored prairies, and there's less stored soil carbon in restored prairies than in a nearby remnant. And the soil carbon is actually very significantly less. It's almost 40% less in this restoration than in a remnant. So fundamentally, what's changed is that potentially these soil microbial communities that were adapted to carbon storage and carbon sequestration have shifted the ecosystem from being one that is a carbon sink to a carbon source, meaning restorations are sources of carbon dioxide when they could be sinks for carbon. They could be preventing the production of that greenhouse gas with proper restoration techniques, but with traditional restoration techniques that focus on plants, they may not be recovering as much function as they could be with some of that focus. So the second reason why we might care about soil microbial communities and prairie conservation corridors is because they provide a habitat for late successional plants. One of the first reason I talked about that we might plant a conservation corridor is to increase plant diversity in the ecosystem and to provide habitat. So if we're going to have this corridor have high diversity for a long period of time, that means that not just early successional plants, which are the ones that we see here, those species that establish first and establish well, not just those plants, but those that are adapted to a really robust prairie ecosystem that might develop later when there is this change in soil carbon or change in soil microbial community composition. These later successional plants also need to develop. Otherwise, a lot of times when I talk to our land manager partners, they wonder about why these corridors or these restorations end up becoming predominated by grasses. And part of that is because grasses are a bit more competitive, especially when there's drought, than some other types of plants. So you don't want to end up planting a prairie conservation corridor just to see it become a monoculture of a few different things of grasses, right? So you don't just want to see a grassy habitat, you want to see a fully diverse community. So we conducted a study last year with my ecology class. This is sort of a preliminary result, just seeing whether late successional plants grown in agricultural soil can germinate. And this graph is showing seedling growth, and in red, if you see more red, that means there was less growth of 16 different seedlings that were planted of each of these different plants. And so you can see there's reasonably good growth of the early successional plants. You know, some of them didn't grow too well, but most of them grew pretty well. But when you look at the late successional plants, you see really a lot of red, right? So there's not much seedling growth for most of those late successional plants. For the ones sort of mid-successional, which I might argue it's also early successional plant that grew well, is Andropogon giardiae, which is that big blue stem of grass. So this is potentially the reason why we see more grasses developing in these systems that are planted, and eventually they shift away from the early successional species and become these grassy systems without too many late successional flowering plants. And part of that might be this reason. So late successional plants are super picky. They don't like ecosystems that don't have the right type of associates, and they won't establish in those soils. So it's a really nice body of work that's been done by a group in Kansas, Koziel and Beaver's work, that is looking at the specificity of late successional plants towards certain types of particularly mycorrhizal fungi. And they've developed actually a business of cultivating mycorrhizal fungi and then determining whether certain organisms or certain plants require a certain specific set of mycorrhizae. And what they found is that it goes down to actually even being locally sourced mycorrhizae are required for some plants to establish in some soils. You have to get the mycorrhizae from that region. It won't even work if you get mycorrhizae from a different region, but it's the same species. So these locally sourced fungi are really potentially important to getting late successional plants to establish. Early successional plants, it seems like they don't care as much in terms of which fungi they associate with. But these late successional ones won't develop unless they have the right ones. When it comes to bacteria, very little is known about bacteria or other soil microbial community members that might be present. And in general, we don't really know whether those organisms that form indirect relationships, that form the other composition of the community surrounding the root structure of the plant that aren't directly associated. We don't really know whether there are certain specificities of those that are required for late successional plants to establish. And that's one thing we're really interested in doing, is starting to parse out, well, what community players need to be present in order to get these late successional plants to establish and establish well. So those are two good reasons. I'm sure there are other reasons as well for caring about soil microbial communities in prairie conservation corridors. So far, I've hopefully convinced you that prairie conservation corridors benefit agricultural systems, but they also require an investment from the farmer. Soil microbial communities don't recover very quickly or at all from agricultural use. Recovering soil microbial biodiversity will improve ecosystem functions associated with prairie conservation corridors, such as carbon storage and habitat for late successional plants. But how do we do this? What are some methods that we can use to start to develop this type of integrated above ground, below ground management strategy? So that's what I'm going to get into for the very last part of my talk. Here are some strategies for restoring soil microbial communities and paths to success, and then some of the work that's come out of my group. But I want to pause here and see if there are any questions at this point.
Yes, we do have quite a few questions. So related back to corridors, when we were talking about that, to be effective for the agriculture, do corridors need to be rotated? Jackie was asking this.
So do you mean, I guess, I'm gonna guess, because I can't interact, but I'm gonna guess you mean, does it need to be shifted over every few years and then
I'll allow her to talk and answer if she wants to.
Okay. I think what she means is does the corridor need to be moved over? So there's a few different ways to think about this. So the first way to think about it is we want to place the corridor in a place where it's going to have the maximum benefit, and maybe that's in a downslope. So you wouldn't want to move it from there if it's providing the maximum benefit there to prevent erosion. On the other hand, if you have a corridor present for a while, it's going to improve the soil health and potentially make it a little bit better of a habitat for crop plants. So in that case, moving it would be beneficial. The farmers that work at the Edward Lowe Foundation actually hay the prairie conservation corridors and then use it as hay. So in that sense, it is actually beneficial for their the overall management of their farm. But they haven't moved them yet. So I think you can think of it in a couple different ways. It depends on where you've placed it, right? So if it's placed in a place where it is actively preventing erosion, I'd say don't move it.
Okay, so another question we had was, have there been experiments to see if soil transplants from remnant soil areas to previously cultivated and disturbed soil areas can speed restoration?
I really hope this is my grad student, Zach.
It's not, it's Kenneth.
Oh, that's too bad. Well, I'm going to get into that next, Kevin. So yes, there have been.
Good job. You already got ahead of us. Ricky asks, could herbicide use that potentially kills local fungi and microbial communities be detrimental when used to clear an area for prairie restoration?
Yeah, that's a really good question. We So when we established this, we worked with the Edward Lowe Foundation to establish this corridor. We didn't use pesticides. We didn't want to have that as a sort of a covariant. We have thought about using pesticides. We've also thought about using herbivores to graze it. So get some goats in there, cows to graze. But that would be something that I would be concerned about, right? So establishing an experiment, I didn't want a pesticide near it because it could potentially disrupt the types of fungi that are present in the soil. And one of the things I'm going to get to is whether those are present or not in the soil. And the idea is that potentially they are there and we wouldn't want to kill them.
And one other question, and then we'll need to keep going, but we'll get to the rest of your questions at the end. What % of natural prairies still exist in the US today?
Oh, the estimate is that it's been reduced from anywhere between 89 to 99%. So this reduction, depending on, it's usually by state or by county. So in Kansas, the Kanza Prairie is a big preserve of prairie, so there's less reduction of prairie in Kansas than in other places. But as you saw in that map in Iowa, they're almost completely gone.
All right, let's keep going.
All right, so how do we do this? Strategies for restoring soil microbial communities. I see there being two possible ways of going forward. And we've done some experiments to demonstrate whether or not they work. So the first approach is to use what I call the probiotic approach, which is what Kevin's question was getting to, which is, can we inoculate microbial communities into a corridor and improve the soil microbial community there with maybe these remnant prairie microbes? If you think about a probiotic, you usually think about yogurt. So if you have a disrupted gut microbiome, you might eat yogurt to help buffer that microbiome and provide some beneficial microbes to your gut ecosystem. This is sort of the gut microbiome management of agricultural soils is this yogurt addition. And we're not adding yogurt, we're gonna add the right type of microbes. We're gonna add remnant prairie microbes to these soils. The other approach is, again, using this gut theory, is that we could use a prebiotic approach, which adds a substrate that slower growing microorganisms would preferentially use. So in this case, we're thinking about you taking your benefiber. You're gonna take your fiber pill. You're not actually taking a live microorganism in this case. You're taking a supplement that will feed the organisms that are hopefully already in your gut and help them to become more abundant in that microbiome. So you take a probiotic or a prebiotic approach, and we have tried both. So the probiotic approach, we attempted a couple of times. We added remnant prairie soil microbial inocula, so we did cellular extractions. We also added soil to experiments in both the greenhouse and field experiments. And the greenhouse experiment, it was with plants of different numbers of plant species. In the field experiment, it was in these 1 by 1 meter type of plots where we added inoculates of remnant prairie soil microbial communities. And I'm not going to go into the details because what we found is that there was no increase in the total amount of fungi, which is what we hope to see. There was no increase in the bacterial oligotrophs, which Microbial and acidobacteria didn't increase. There weren't any changes actually in the bacterial or fungal communities. There wasn't any change in plant biomass yield in the greenhouse experiments. And in fact, this approach, which has been proposed a lot of times, also requires disrupting remnant a bit, which is frowned upon and also isn't really that tractable if you're thinking of an entire prairie conservation corridor where you might have to inoculate a whole several acres of land area with some sort of remnant source. You wouldn't want to disrupt a remnant that much to be able to expand to that size. So why didn't this help at all? And this has been an idea that's been floated a lot of times, either by land managers or in different research groups. We think that what might be happening, at least in our soils, is that slow-growing microorganisms, these oligotrophs that are added to this agricultural soil, are just really maybe rapidly outcompeted by fast-growing soil microorganisms that are already present in the soil, the copiotrophs that are in the agricultural soil. So adding them, they're just gonna lose that competitive battle with those other organisms, and you've also disrupted some remnant or gotten some soil from a remnant to do a cellular extraction, and you know, maybe it's not going to benefit the system that much. So we say no, that might not work, and it's maybe not the most feasible approach anyway on a large scale. There are some studies. I saw, I don't know if they've published it yet, but there's this group in Kansas that have been doing this in a different soil type, and they do find that adding remnant soil microbes did seem to provide some benefit to plant establishment. They also are the group that is showing that specific types of fungi seem to be required. So it might be more appropriate to culture specific types of fungi for the specific types of plants you want to grow and then inoculate those into the soil. But a bulk addition of remnant soil or a bulk addition of a cellular extract from remnants didn't seem to work for us. So the prebiotic approach, this fiber approach, is going to assume that some oligotrophic microbes are already present in the agricultural soil, and they're just in low abundance and they need the right food source. And one cool thing that microorganisms can do is either they survive in very low abundances in some cases and then can flourish when the right food source is present, or they can become dormant and stay in that dormant state until conditions become right for them to be competitively at an advantage. So we tried this type of an experiment a couple of different ways. The first way was with a greenhouse experiment where we added the most basic form of fiber, cellulose. So it's a cellulose salt to our potted soil plants. And we looked at bacterial communities and fungal communities over time with these different plant communities. And what we found was really promising was in the cellulose treatments, the total lipid biomass, which is the total amount of microbes basically in the soil, increased by almost 300% with cellulose addition. So it really stimulated production of microbial biomass, which if you convert that into soil carbon storage, it's increasing the amount of soil microbial carbon. We also looked at whether that biomass was related to bacteria or fungi, and what we saw was that it's actually related to fungi. So we're seeing an increase in the types of organisms that are going to produce more cellular biomass, fungi, when you add cellulose. And when we looked at the bacterial community itself, we also saw that it increased the total number of oligotrophic bacteria, and the relative abundances of acidobacteria and verrucomicrobia both increased, those two oligotrophic bacterial phyla. It also decreased one of the copiotrophic phyla, actinobacteria, with addition of cellulose. So all of this is promising. And we thought it was promising enough to attempt it again in a field experiment where things can go much more awry than in a greenhouse experiment. So we conducted a field experiment. We started it in April 2019, where we added both cellulose, this sort of crystalline form of cellulose, as well as dried plant biomass as two different treatments to these field plots. And so why did we do this? Again, cellulose is going to be expensive if you buy it in this crystalline form. And if you're going to do it on a multi-acre scale of an entire conservation corridor, it's probably not very feasible for most farmers or land managers. But what might be feasible is to mow some prairie plant biomass and grind it up and then add that into the field or till it into the field if it's a till system before conducting the seed planting and restoration. So this is what we tested and we're currently testing at both two different soil types. So on the left here is the cellulose addition. This cellulose slurry was evenly spread across these 1 by 1 meter plots. And then we also tested a biomass addition. So this is the biomass being added to these 1 by 1 meter plots. And in this case, it's just little blue stem biomass, but we've also tested it with mixed plant biomass, which actually might provide more diversity of biomass substrates. One thing that is really kind of crucial to think about is that different soil types have different microbial communities and may respond differently to these types of additions. So we are actually doing this experiment in 2 different soil types, one that's pretty sandy and one that's much more like clay. And as I said, we started this in April 2019. We took our first samples in August 2019, and then hopefully we will be taking our second set of samples in August 2020 with some restrictions on fieldwork right now. We're not sure, but that is the plan. But what we saw was really pretty promising, was that in the sandy soil, this is one of these ordination plots where the points that are closer together mean that the communities are more similar to each other, and the points that are further apart mean that those communities are more different from each other. So with biomass addition in the sandy soil, what we saw is that the community is actually shifting within 3 months away from what is seen in the control plot where we didn't add anything. And the shift is actually towards these more oligotrophic phyla, particularly Verruca microbia. So this is promising. It's happening within 3 months. Whether or not that continues and becomes more and more distinct is something that our next set of sampling will tell us. But it certainly seems interesting that this is happening in 3 months instead of 3 decades. In the silty clay soil, so in this much more moist, finer soil, we saw something pretty different, which is interesting as well, is that there weren't really any differences in any of our treatments, but we seem to see more of a response with just that type of cellulose addition as opposed to the biomass addition. This could be related to a lot of things. It could be related to a drought that occurred. It could be it may just be related to the timing of the sampling. And so again, it'll be exciting to see what we see in August 2020, hopefully, whether these 2 communities have converged in these 2 different soil types, or whether it really does matter whether we add cellulose as a salt in one type of soil versus biomass is just fine in a sandier soil. And this will really inform whether this type of approach can work and lead to more soil carbon storage and potentially a interesting management strategy that could be employed. So we might be seeing these different prebiotic responses in the soil type, but in either case, it seems like the prebiotic response is interesting. So I just want to point out that these results look promising, but there's still many unanswered questions. Do communities changes increase over time? Does fungal biomass increase over time? Does CO2 emission decrease over time? Can we establish late successional plants earlier or later? And then overall, how are these plant microbe interactions influenced by these types of approaches? So with that, I just want to point out that I really think prairie conservation corridors are a neat management strategy that improve agricultural ecosystems. I really like them just the way they are, but I think there's potential to also make that investment even more beneficial by understanding more about soil microbial management strategies. And that's really what we're focused on doing, and where my research will be going in the next several years. So thank you all so much for being here. And I want to point out that a lot of this work was done by my grad student, Zach Whitaker, who is in the yellow circle there, as well as several undergraduate researchers in my group. We collaborate with the Edward Lowe Foundation and the Washington Conservation District in Minnesota to do this biomass addition project. And my collaborators as well have been really helpful in helping to figure out these where this project is going and start to think about the next steps. And also thanks to the Pierce Cedar Creek Institute for hosting all of this talk and everyone being here. So with that, I will see if there are any further questions. If you don't feel like asking me a question now, you can always email me. That is my email address. And you're welcome to email if you have a question or a comment.
Thank you, Kathryn, very much. And I understand we are going past the time a little bit. So if you do need to head out, feel free to do so, but we will try to get to your questions now if you have time to stay. Otherwise, this will also be up on our YouTube channel as well as our Facebook page. So if you do need to go, feel free to go. So we did have a couple of questions about your dried plant biomass. One was, do you mean composted biomass? And also, what are your thoughts on hot spring burning and management that removes the dry biomass?
So the biomass was not compost, it was just dried biomass.
Okay. And your thoughts on hot spring burning and management that removes the dry biomass?
Yeah, so with this strategy, the one that I'm describing, I think the biomass would actually need to be tilled into the soil. So this wouldn't work for a system that's been no-till. I've actually had this conversation with some of the folks out at Kellogg Biological Station where they have no-till systems, and they're not a fan of this idea because it would have to require them tilling the system. But if it was a tilled system, you'd till in this biomass potentially, and then it would actually be buried under the soil. So spring burning is maybe not going to affect that either way because it's not tilled into the soil. I think burning is important to do and reduces, it improves the amount of carbon substrates that end up in the soil, but it also improves the protection of the conservation corridor against invasives. So I wouldn't say get rid of burning or get rid of that litter, but I would say put in and till in initial biomass at the time of restoration. That said, I want to take my samples in August and see whether this has actually worked.
Awesome. Yeah, to be determined, right?
Yeah.
There was a question, you referred to proper restoration techniques that could turn a prairie restoration into a carbon sink. Somebody was wondering if you could elaborate on those proper restoration techniques.
So probably proper isn't the right word, and I apologize if that offended anybody. Integrated above ground, below ground restoration techniques.
Awesome. Another question we had was, how organic is organic produce if only 3 years of restoration is practiced prior to the land qualifying for organic certification?
Oh, that is a great question, and I'm not sure. Yeah, I guess if you're using, so it would depend on a lot of different things, I think, but I think if you're using plant biomass as your source of compost and fertilizer, it eventually becomes an organic system. But yeah, unfortunately, I don't know the answer to that one.
Okay, we can't know it all, right? Corrine was asking, are the locally adapted fungi that late successional plants need already present in the soil during the early successional period? Or does the pool of these fungi species develop during early succession? Or will it be necessary to add these species in manually during later succession?
Is this Corinne?
Yes.
Hi, Corinne. So what Cosio and Beaver are showing is that they needed to add it, right? So they needed to add the particular fungal species to these particular late successional plants, and that it depended on which fungal species it is. So I think that we would need to do this experiment for a longer period of time to know whether that's the case and really understand more about not just the fungal associations, those direct associations, but also what characteristics of the soil community in general are present, so and are changing over time with these below ground restoration strategies. There is potential that they're present, and there's potential that they could be selected for by either planting seedlings of the plant or providing the right prebiotic substrate for them, and it may take some work to figure out what's that right prebiotic substrate. but it may also be important to inoculate. So I think it will depend very specifically on which plants are of interest and if they are specialists for these fungi.
That makes sense. Matt was asking, what are some similarities or differences to microbe restoration in forested ecosystems? Does restoration follow a similar pattern as in prairie systems?
That's a great question. And there's less known about that, I think, than in prairie systems, just because so much work has been done in these sort of agricultural and prairie systems. One of the places we worked for a little while was in oak savanna, which is much more similar to a prairie than a forest, but it also seems that some of these trends hold true for oak savanna systems. So I wouldn't be surprised if there could be ways to integrate above ground and below ground restoration in forested systems, but also it's going to be much longer term potentially because of the length of time for those plants to establish. Actually, Ellen may know the answer to this more than I do.
I was just going to say, I know on some research that I did when I was a graduate student down in Costa Rica, there were some forested systems that never got the same microbial community back, even after decades of being in restoration. It was just always going to be a different microbial community. We were focusing on mycorrhizal fungi. It didn't mean that they were less productive. It was just that it was a completely different system. So different stuff that was there. Yeah. A couple more questions. For those that are wanting to get prairie plots started in their yard, would adding mulch or other dead plant materials be a suggestion that might help prairie plants and their microbes be more competitive against the more disturbance adapted species, for example, like dandelion? Sarah's asking this.
Oh my gosh, yes. So this, in addition to photography, is sort of my other pastime, is trying to get native prairie plants to establish in my yard. So I just mulch with, so I cut down the dead senesced plant material every fall, and then mulch it up and then put it into the garden. I don't add like store-bought mulch. I use that as the mulch, and it's been extremely successful. So that rattlesnake master is traditionally pretty difficult to cultivate, and there's a bunch of it coming up. There's all these kind of later successional plant species that are pretty well established at this point. So it's been great to see, and even just mowing some grasses and putting that into your mulch is going to prevent weedy species. For the first couple of years, I had to actively weed out things, but after that, I mean, I don't think I spend much more than a couple days a summer weeding that sort of prairie plant plot. So definitely mulching with the native plant material seems to be very helpful. Part of that is because as it's decomposing, it's selecting for the microbes that might be associated with that plant. So the particular type of carbon substrates that are produced as it's decomposing selects for the microbes that might benefit the next season.
Awesome. Just a couple more questions. Can a city like Detroit, with areas where houses have been removed due to urban blight, convert to those areas? Can we convert those areas into prairies?
I would say go for it. I don't know who owns those properties, but yes. Honestly, putting these little prairie plantations into urban areas is a great idea. Kalamazoo has been really proactive about that. There's a lot of little spots of prairie plants and native plantings throughout the city. If anybody is ever riding along the Kalamazoo River Valley Trail, you can see this whole excellent area of native plantations of mostly milkweed and other types of butterfly attractants. So yeah, integrating native plants into suburban and urban ecosystems is a neat idea, and I think it's extremely important for some of those, especially long distance migrators like monarch butterflies, for example, to have that habitat be in urban ecosystems. Otherwise, they go through this urban desert of not having a particular source of food or area to reproduce, and really can put that type of species at risk.
Great. And just to reiterate, make sure people got it. So if we do plant prairies, especially in some of these urban areas, will the good soil guys just show up?
Well, what we are seeing is maybe they're already there and they just need to be encouraged. So the plants will likely establish given enough care and mulch. It's a matter of whether they will be really prolific and whether the late successional species might develop. Your early successional species are going to provide a lot of ecosystem benefits, though, don't get me wrong. So planting a bunch of coreopsis in this coneflower, for example, or sunflower, false sunflower, black-eyed Susan, or cup plant. All of those are early successional species that are going to benefit an ecosystem. So even if the microbes aren't going to be present or support, maybe some of these especially late successional species, it's still a good idea to have those native plants in urban environment.
Awesome. Thank you, Kathryn, so much. I think we got all the questions. We really appreciate your time. Oh, and by the way, our native plant sale, if you want to get some of these native plants, is going to be June, oh boy, I forget the date right now, June 13th, I believe. It's a Saturday. But if you want to get some of these plants, you need to pre-register, or you need to pre-order, the plants by June 1st. So we will have a lot of these native prairie plants. So I guess closing thoughts, go plant a prairie, right?
Yes, and know what's native to your native ecosystem. So I know that most people in this session are from Michigan, but if you're from someplace else, take a look at some websites. The Monarch Way Station website or the Monarch Watch Way Station website is a great one for figuring out what types of native plants attract butterflies. But there are likely plant sales in your area as well that are going to have native plants.
Awesome. Well, thank you so much. We appreciate your time, and thank you everyone for joining us today.
Thanks, everyone. Thanks, Ellen.
Yep, bye.